Hi, we are going to talk about the sensorimotor systems. First, a question. Why do animals need a nervous system? But first, we have to, before answering this question, we have to look at the body components. First, the body needs nutrients, energy, and oxygen. And this is the way of taking these nutrients and oxygen is through the digestive and respiratory apparatus, the digestive apparatus and the respiratory system. So then this goes to the vascular system. From the vascular system, this has to be filtrated. The blood has to be filtrated. We have to reproduce. And for that, we need a genital component. So we need a genitourinary apparatus. These are viscera. To filtrate blood, we need a urinary component. And this, again, it forms part of the genitourinary apparatus. To stand and move, we need a locomotor apparatus formed by bones, joints, and muscles. And everything is controlled by the nervous system. So the answer is we need a nervous system to be able to move. So then, why animals need a brain? The primary purpose of a brain is to move around in the environment. For that, the animals need intelligence. They need to interpreting sensations, to initiate voluntary body movements, and controlling all the behaviors. And for that, we need afferences from the inside and outside the body. We need to integrate these sensations. We need memories, because some situations in the past may condition our movements and our future situations. We need will. We want to go. We want to do things. We need thoughts. And everything is controlled under afferences. So here you have a summary of the parts of the central nervous system. The central nervous system is formed by a cerebrum, a cerebellum, a brainstem, and a spinal cord. And each part has its own function. The cerebrum, the function is participates in will, thoughts, sensory, integration of sensations, memory. All this takes part in behavior. And also the cerebrum participates in the initiation of skilled movements. You know, in the prosencephalon, we include the cerebral hemispheres and the rostral portion of the brainstem. The thalamus is included there. So the cortex, the basal nuclei and thalami are related in the function of modulation of fine movements. The cerebellum for movement coordination. The brainstem controls balance against gravity, controls automatic movements. It participates in less skilled movements, in reflexes, and to keep the homeostasis. And finally, the spinal cord that participates in reflexes and the transmission of information towards the encephalon and from the encephalon. So movement control, we need planning. We have to plan this movement. We need a prediction, what's going to happen. And we need to correct misadjustments. So there are control pattern generations, CPGs, to control this movement. For reflexes, for basic activities, as eating, breathing, fight, and basic locomotions, control pattern generations are important centers to control these activities. The cerebral cortex acts on brainstem control pattern generations only if fine or visual motor coordination is needed. So cerebral cortex has a highest control. For it is important when locomotion is in unfamiliar locations. So the cerebral cortex acts on control pattern generations of the brainstem that in turn, they act on control pattern generations of the spinal cord. Lesions in the cerebral cortex affect sensory information and fine movements, but not basic locomotion and basic movements. It has been proposed that the walking therapy is useful reactivating control pattern generations after a mild spinal cord injury. Here is a drawing representing different centers that control movement. So we have the accumbens that is connected with the limbic system. You know, this is a basal nuclei, and it's important because it's related with rewarding of the situations. The cerebral cortex for skill movements. Yeah. The basal nuclei, the rest of the basal nuclei, programming the execution of movements and also participating in behavior. So lesions that affect these basal nuclei may present with compulsive movements of unwanted movements. The thalamus is a center of integration of all these sensations. We have the subthalamus, very important, the subthalamus. We have studied the subthalamus when we were studying the brainstem, and it's needed for initiation of the voluntary movements. Sorry. We have the substantia nigra. You know the substantia nigra has a function in regulating the activity of the basal nuclei. We have centers in the mesencephalon, the cuneiform nucleus located in the cerebral peduncles, or the pedunculopontine tegmental nuclei. The first one elicits movement, and the second suppresses muscle tone via the reticular formation. These are very important centers to initiate movement. If they are stimulated by higher centers, they have to be stimulated, these ones. We have the locus coeruleus that participates in activating muscle tone. We also have parts of the cerebellum, the fastigial nucleus, that projects to the brainstem and controls the muscle tone. And also is participating in the correction of error correction mechanisms in visual pathways, right? We have the vestibular nuclei to control the postural muscle tone and the reticular formation, pontine and medullary reticular formation, that controls asymmetrical postural reflexes by regulating different types of muscles. If in a decerebrate cat, depending on the site of the lesion, we may find different results. If the lesion is rostral to the subthalamus, we see that spontaneously initiate movement with well-coordinated posture control. So the animal can initiate a movement if the lesion does not affect the subthalamic nuclei. However, if the lesion is behind the subthalamus, to initiate the movement, these centers here, this cuneiform nucleus and the pedunculopontine tegmental nucleus, need to be chemically or electrically activated, right? However, in both situations, this walking, it's neither directed nor adapted to the environment. So even the movement is spontaneously produced, cannot be controlled voluntarily, because for that we need the prosencephalon, right? In particular, the cerebral cortex for control of voluntary movements. So here is the summary. Sensory signal that could be from proprioceptors coming from the interior or exteroceptors coming from the external of the body. This sensory signal is transduced to the central nervous system. And in here we need the memory, because there are learning situations and the memory may help us to control the movement, or also in unfamiliar situations to remember previous situations and to prevent failures. So once this information has been processed, we develop a movement that could be voluntary or involuntary. And the components of this movement could be cognitive or emotional. In the case of cognitive, we need the cerebral cortex. In the case of emotional, the limbic system takes an important role. Both components end in the brainstem and in the spinal cord to act on motor neurons. So, a stimuli is captured by the peripheral nervous system and it is transported to the central nervous system. We call it sensory. And from the central nervous system, the stimulus and is transported back to the peripheral nervous system. We call it motor. The first one is the afferent pathway, and the second one is the efferent pathway. So once it gets to the peripheral nervous system, we develop a motor activity. And this motor pathway may involve pyramidal and extrapyramidal motor systems. Another important thing is the different types of movements. There are skill movement and less skill movements, right? For less skill movements, locomotion, basic locomotion, eating, etc. The pyramidal system participates in skill movements, whereas the extrapyramidal system participates in less skill movements. And what is the difference? The pyramidal system uses direct connections. There are just one neuron that comes down to an interneuron that synapses on the motor neuron. So there is a direct connection. There is not a losing of information. The information is not loose in this pathway. However, for the less skilled movements, the higher centers are acting on motor neurons through a series of neurons. There are several connections in between. So, again, here is a summary that what I want to point out here is that the inhibitory motor function is very important for an accurate motor control. It's like a car. When driving a car, it is as important or even much important to have a good brake system than an accelerator. So, as you see, whereas the corticospinal tract that acts on alpha and gamma motor neurons, that facilitates distant flexors and inhibitory the distant flexors, or the red nucleus through the rubrospinal tract facilitate flexors and inhibit extensors, and the medullary reticular formation that inhibits extensors. So the inhibitory function is very, very important. So when the lesion affects the cerebral cortex or adjacent internal capsule or the basal nuclei, here you see the basal nuclei, the cortex, the internal capsule, right? So the gate generation, it is not impeded. This is what De La Hanta says. However, so it is because these gait movements are movements that are not skill movements. However, the result is a motor impairment due to the lack of sensory processing because the sensations they are not correctly processed at the cerebral cortex. So the motor function is not properly, or it is not correct. However, the animal can move with or can perform less skilled movements. So we can summarize that a deficit in general proprioception plus preserved motor activity for, I mean here, for not skilled movements, the lesion should be located at the level of the prosencephalon, okay? If there is a deficit in general proprioception plus a motor impairment, so the lesion should be caudal to the prosencephalon, okay? Because at the brainstem, caudal to the subthalamus, involving the subthalamic area, mesencephalus, and reticular formation of the brainstem, here are centers that are involved in movement, however, but not fine movements, okay? So, the neurological pathways in the central nervous system are of two types, sensory for afferent and motor for efferent. We'll study the general proprioception first. This is the position sense, okay? Well, it could be for static position or for kinesthesia in movement. Right. Proprioceptors that are located in the musculoskeletal apparatus, they are sending information to the spinal cord. At the lumbar level, the intrafusal receptors and the Golgi tendon neurons, they send myelinated fibers to the spinal cord, to the dorsal horn. They end in lamina 5, 6, and 7 of this spinal cord. Then they synapse there in what is named the thoracic nucleus. This is a long column of cells that extends from T1 up to L4. Then second order neurons go to the lateral funiculus and they form the dorsal spinocerebellar tract that enters the cerebellum through the caudal cerebellar peduncle. Fibers coming from Golgi tendon neurons, they reach the spinal cord, okay? They are formed by myelinated fibers. They need to be fast, this information needs to be fast. And they reach lamina 7, formed by a lot of interneurons, in the dorsal horn of the spinal cord. Second order neurons cross the midline through the white commissure and form part in the lateral funiculus of the ventral spinocerebellar tract that reaches the cerebellum through the rostral cerebellar peduncle. And once inside this cerebellar hemisphere, they cross again the midline and they end up in the ipsilateral cerebellar hemisphere. For the forelimb, fibers from the Golgi tendon neurons are also myelinated fibers that enter the spinal cord, and they reach a column of cells that is located at the more ventral portion of the dorsal horn. And here you have, this is and they extend from C1 up to C8. Second order neurons then, they are projected to the lateral funiculus, and they form the cranial spinocerebellar tract that is located medial to the ventral spinocerebellar tract. These axons, these neurons, enter the ipsilateral cerebellum through the rostral cerebellar peduncle. For intrafusal fibers, these intrafusal fibers enter the spinal cord, okay? They enter the spinal cord as myelinated fibers, but they do not end in the dorsal horn, okay? They go straight to the dorsal funiculus. And in the dorsal funiculus, they form the cuneate fascicle that reaches the lateral cuneate nucleus that is located in the medulla oblongata. And second order neurons are projected to the ipsilateral cerebellum through the caudal cerebellar peduncle. So in the end, the cerebellum that receives the information is the ipsilateral cerebellum. So this information, the unconscious proprioception, is going to travel through the cuneate fascicle in the dorsal funiculus, dorsal spinocerebellar tract, and ventral spinocerebellar tract. But for some aesthetic conscious, or for the conscious proprioception, okay, the pathways are going to be different. In the case of the hind limb, for the pelvic limbs, myelinated fibers enter the spinal cord. The receptors are the same, okay, and the fibers are the same. So, but once inside, they take different pathways. These ones from the pelvic limbs enter the dorsal funiculus, and they form in the dorsal funiculus the grathial fascicle. And this grathial fascicle has to go up until up to the medulla oblongata, to the nucleus gracilis. Okay. However, most of the fibers that form the grathial fascicle abandon the grathial fascicle at the thoracolumbar level to make synapses in lamina 7 of the spinal cord in a column of cells called thoracic nucleus that extends from T1 up to L4. Second order neurons then form part of the lateral funiculus, incorporate to the dorsal spinocerebellar tract, and they go up until the medulla oblongata, reaching the Z nucleus that is dorsomedially located to the nucleus gracilis. So as you see, up to 90% of the fibers that form the grathial fascicle abandon the dorsal funiculus at the thoracolumbar level. So as we will see in histology, the dorsal funiculus will become thinner at the thoracolumbar level. Okay? And very few fibers reach the grathial nucleus. For the forelimb, myelinated fibers enter the spinal cord, but they do not enter at the level of the dorsal horn. They just continue in the dorsal funiculus, forming the cuneate fascicle up to the medial cuneate nucleus. Okay? The medial cuneate nucleus is located medially to the lateral that we have seen in the unconscious proprioception. From these three nuclei, the neurons that come from these nuclei project their axons to cross the midline, and they form the medial lemniscus up to the thalamus, and from there to the somesthetic cortex. So for conscious proprioception is the contralateral cerebral hemisphere that is in form. For unconscious is the ipsilateral cerebellum. For conscious is the contralateral cerebral hemisphere. Here, what are the pathways involved? The dorsal spinocerebellar tract and the cuneate fascicle and brachial fascicle. So we go to histology now. Here you have a dorsal view of the spinal cord at the lumbar intumescence. Look the dorsal funiculus. Here we have the roots, or the dorsal roots of the spinal nerves. So you have a very big or very wide dorsal funiculus. In histology, in a Bielczewski, you see this dorsal funiculus, and it's formed by the brachial fascicle. Remember that the brachial fascicle is taking conscious proprioception from the hind limb. However, if we move to the thoracolumbar region, we identify that the dorsal funiculus gets smaller. It is much smaller. The brachial fascicle is losing fibers because most of the fibers abandon the brachial fascicle in order to reach the thoracic nucleus. And from there, second order neurons are going to incorporate to the lateral funiculus, forming and going up to the Z nucleus. If we move at the cervical level, we see that at the cervical intumescence and up to the first cervical spinal segment, the dorsal funiculus gets wider again. You see, it's bigger because even though the brachial fascicle has become very small, we have added another fascicle, that is the cuneate fascicle, because this cuneate fascicle is taking myelinated fibers that enter in the spinal cord from the forelimb, and they reach the medial, the medial cuneate nucleus. Okay, so we can see that morphologically. Okay, so now we are at the level of the medulla oblongata, and what do we see here? In a brain stem, we see a very thin brachial fascicle here, a very big cuneate fascicle. This is the spinal tract of the trigeminal nerve, and laterally we have the spinocerebellar tracts. Okay, these are and this is the caudal cerebellar peduncle. So on histology, we identify here the brachial nucleus, the medial cuneate nucleus, and the lateral cuneate nucleus. In hematocylin eosin, we also see that. On top, very on top, we have the brachial fascicle. Okay, underneath we have the brachial nucleus. Here on top, we have the cuneate fascicle, and underneath we have the medial cuneate nucleus. So the structures that we see dorsally on the brain stem, they are fascicles here. So the brachial fascicle, cuneate fascicle. If we go a bit up, we can identify this area, and we are going to enlarge it. And you see, this is the brachial nucleus and the medial cuneate nucleus, and we see an extension of this brachial nucleus dorsomedially. This is the Z nucleus. So in these images, this is a transverse section at the level of the decussation of the pyramids, okay? And we also have the brachial, cuneate, fascicle, brachial fascicle, cuneate fascicle. We go on histology, and again, this blue square is enlarged here, and we see the Z nucleus dorsally located. This is the brachial fascicle, all right? The cuneate fascicle. So ventrally, we have the brachial nucleus. And in this red square, enlarged here, we have the nucleus cuneatus medialis and the nucleus cuneatus lateralis. And on top, we have the cuneate fascicle. This structure here, all these axons that have been cut transversally. So we want to identify the thoracic nucleus, and it's easy. You see, the thoracic nucleus is located in lamina 7 of the spinal cord. Here it is. And you see, do you see the thoracic nucleus extending from T1 up to L4? Right here. Or in Bielczewski, silver impregnation, right? This is the Clark's nucleus or thoracic nucleus. So this nucleus is receiving unconscious proprioception from the dorsal root fibers. It receives conscious information, or that is conscious proprioception, from the dorsal funiculus, okay? And is sending second-order neurons to the ipsilateral lateral funiculus and to the contralateral lateral funiculus. Let's have a look. We go to histology again, and here we have the dorsal funiculus, the dorsal horn, the central canal, and the homologous dorsal horn. Okay, you see fibers that are entering, right, dorsal to the dorsal horn. These fibers reach the thoracic nucleus from the dorsal root, because the fibers that have to reach the thoracic nucleus, that is located at this level, they don't go through all the gray matter. They go on top of the gray matter, and when they reach their site, they enter. And also here you have fibers. This is enlarged here. These fibers are fibers that enter the thoracic nucleus from the dorsal funiculus. This is for conscious proprioception. These ones are for unconscious proprioception. Again, we see this is transverse section, right? We also see the spinocervicothalamic tract located at this level, lateral to the dorsal horn. But we are going to focus in here, the thoracic nucleus. You see, this collection of cells with a lot of axons is a column. This is the Clark's nucleus. So, as you see, there are fibers leaving the thoracic nucleus, and they go to the lateral funiculus, okay, to form these spinocerebellar tracts in particular, okay. The ones that go ipsilaterally form the dorsal spinocerebellar tracts. The ones that go contralaterally form the ventral spinocerebellar tracts. And we also identify those ones here, right? Fibers that from the dorsal root, enter the laminae of the dorsal horn. They go to the lamina of the dorsal horn in order to reach the thoracic nucleus. These are for unconscious proprioception. This thoracic nucleus changes its location as it approaches the lumbar segments. There you see, right, at T13, right here, the thoracic nucleus. However, at the level of L2, as you see, this nucleus has moved dorsally, right? Dorsal to the central canal, here. And these are the cells that form the thoracic nucleus. So now we are going to talk about a special proprioception. The first we have talked about is general proprioception. Now it's a special. The special proprioception is for equilibrium. The receptors are located in the inner ear, and they are in the cristam polaris or macula that are in the inner ear. So this information is transmitted to the vestibular nuclei, ipsilateral. Remember that these vestibular nuclei are connected with the cerebellum, okay, mostly ipsilateral. From these vestibular nuclei, information is transmitted to the vagal parasympathetic nucleus, and here is this information is related with vomiting. But there are some fibers that form the part of the medial longitudinal fasciculus in order to reach nuclei that control extraocular muscles. And these are important to generate a slow phase of the nystagmus, that the eyes are going to move contrary to the side of rotation of the head. Following the slow phase comes the quick phase with the movement in the same direction of the rotation of the head. Okay, this is the vestibulo-ocular reflex that is important to keep balance. Of course, there is information that is transmitted to the thalamic nuclei and from there to the temporal cortex to be aware of this losing of balance. But to keep balance, we also need vestibulospinal reflex. In this case, there's the vestibular nuclei that have been activated, ipsilateral to the movement, they send information through the vestibulospinal tracts to activate extensors, right? To control the deviation to the side of the rotation. And also to inhibit extensors contralaterally. This is the vestibulospinal reflex. And this information is going to travel through the lateral vestibulospinal tract that is located in the ventral funiculus, but under the ventral horn, and through the medial vestibulospinal tract that is located in the medial funiculus. In the ventral funiculus. The lateral vestibulospinal tract is going to reach almost all the spinal segments, whereas the medial vestibulospinal tract reaches the cervical spinal segments. We have to remember that there is here, medial to the lateral vestibulospinal tract, the spino-vestibular tract. This spinal afferents from C1 up to C4 reach the cervical spinal nucleus in lamina 7, okay? And this spino-vestibular tract reaches the caudal vestibular nucleus, that is to control and to send information from the neck up to the vestibular nuclei. Also, there is, as I have said, from this vestibular nuclei, information is going up to the thalamus. And there are different pathways for the vestibulothalamic connections through the middle longitudinal fasciculus to the central tegmental tract, ascending tract of data, and vestibulothalamic tract. Okay? And these connections are contralateral or bilateral. Something important, or maybe it's nice to remember, that no direct projections from the vestibular nuclei or thalamus have been proved to the hippocampus. Okay? All right. So, then these tracts that are going to come down are the lateral vestibulospinal tract and the medial vestibulospinal tract. Look at here. The lateral vestibulospinal tract is in the ventral funiculus, is ipsilaterally descending, and is sending information to the ipsilateral and contralateral parts of the spinal cord. And these axons are going to end in lamina 7, where there are interneurons, and some of them are direct to lamina 8, where there are motor neurons that are related, or they project to the axial muscles. And through these interneurons that are in lamina 7, information is also activating lamina 9, that is formed by motor neurons that go to appendicular muscles. However, the medial vestibulospinal tract that just reaches cervical spinal segments goes to both sides as well. However, it gets to interneurons of lamina 7 and to motor neurons of lamina 8 to axial muscles. So this medial vestibulospinal tract is not related with appendicular muscles. So there is a summary. The medial vestibulospinal tract, it just reaches the cervical level, and it ends in interneurons of lamina 7 and in motor neurons of lamina 8 for neck muscles, axial muscles. And as you see, the lateral vestibulospinal tract reaches cervical, thoracic, and lumbosacral spinal segments, and ends also in interneurons of lamina 7. Also ends in motor neurons of lamina 8 for neck muscles. But it also ends in lamina 9 that is formed by motor neurons for axial and limb muscles, for appendicular muscles. So the lateral vestibulospinal tract goes to axial muscles, but in particular to appendicular muscles. So, motor systems. For a summary of the motor systems, I think this schema or this scheme is quite interesting. So we have afferent fibers information is getting into the central nervous system. And from there, it travels up to the thalamus and cerebellum. Okay? The cerebellum connects with cerebellar nuclei that project to the thalamus. So the thalamus is informed. The thalamus projects to the sensory cortex, and from there to the basal nuclei and back to the thalamus. And this feedback continues until the information is processed. Okay. So now once the information has been established, this information now is projected to the motor cortex. And then the motor cortex starts the activation. Fibers from the motor cortex come down, forming part of the internal capsule. They go down to the pons. This is the cortico-pontine tract. From the pontine nuclei, information travels to the cerebellum to keep informed the cerebellum, not just for the sensory, but also for the movements we are trying to develop. And the information comes down, activates nuclei of the brainstem as the red nucleus, activates cranial nerve nuclei. This would be the cortico-nuclei tract, activates centers of the medullary reticular formation, and comes down. And then these fibers, as they form the pyramids, they are the pyramidal fibers that activate motor neurons. Okay? Activate motor neurons. And I have put a minus because most of this action, instead of being facilitatory, is inhibitory, is to control the proper movement of the muscle. There are other centers, as you see, the pontine reticular formation, vestibular nuclei that are controlled by the cerebellum. These are facilitatory to extensor muscles, right? Okay. Also, once the red nucleus and medullary reticular formation has been activated, the same information that is going to be facilitatory or inhibitory over the motor neurons. So, as we see, it is difficult to talk about pyramidal or extrapyramidal because at the beginning, at the most rostral portions of the encephalon, the pathways are the same. Okay? So here you have that on the PDFs. For the motor system, from the motor cortex, internal capsule, in between we have the basal nuclei, right? With neural loopings, right? In controlling this activity. And then we could talk about pyramidal or extrapyramidal, okay? Pyramidal, but most of the pyramidal tract fibers end in the brainstem. They do not go down to the spinal cord. Very few go down, okay? So maybe this drawing will help you understand. This is the pyramidal system. Motor neurons come from the motor cortex in the internal capsule. They go down and they end in nuclei of the brainstem, whether they are the mesencephalic tectum and tegmentum, red nucleus, cranial nerves, right? Nuclei, pontine nuclei, the ones here, they go to the cerebellum, they go to reticular formation, and the fibers that continue, but most of the fibers end here. The fibers that continue form the pyramids. Some of the fibers cross at the decussation of the pyramids, and they are gonna go down in the spinal cord. So here is a transverse section of the mesencephalon. All these descending fibers form the crus cerebri in the mesencephalon, forming the corticospinal and corticopontine tracts that they end in the red nucleus, in the tectum, or several areas of the tegmentum mesencephalium. The ones that end in the pontine nuclei, these cells project to the contralateral and cerebellum, entering through the brachium pontis or middle cerebellar peduncle. And the ones that continue form the lateral corticospinal tract or the ventral corticospinal tract. The ones that are in the lateral, there are fibers that have decussate at the decussation of the pyramids, and the ones that are in the ventral funiculus have not decussate. However, they will cross the midline at the point when they enter the gray matter. For the extrapyramidal, what about the extrapyramidal? At the beginning, it's the same. However, the extrapyramidal is formed by all these projections that are coming from the nuclei that have been activated by these cortical fibers. Okay, so the fibers that are coming from the tectum mesencephaly, the tectospinal tract, the fibers that are coming from the red nucleus, structures that have been activated by fibers coming from the cortical areas, from the prosencephalic areas, okay? As you see, these fibers cross the midline at the tegmental decussation, or the point and reticular formation, or the medullary reticular formation, or from the vestibular nuclei. Of course, in this case, the ones that are connected with the cerebellum are coming from the olivary nucleus. So the olivary nucleus is considered an extrapyramidal nucleus. So the pathways coming down in the spinal cord are now different. They are the rubrospinal tract coming from the red nucleus here. These are fibers that have crossed the midline at the decussation, the tegmental decussation. Okay, the reticulospinal tract, the tectospinal tract, and vestibulospinal tracts. These are descending pathways. So, if we compare the rubrospinal tract with the lateral corticospinal tract, or the corticospinal tracts, we see that the lateral corticospinal tract is formed by the fibers of these are going to enter, as you see, the dorsal horn, the intermediate gray substance, and the ventral horn. So the dorsal horn are getting to the 5th and 6th laminae, okay, and to interneurons of lamina 7 right here, okay. These are very important in humans and raccoons, okay. And in these ones, some of them, especially in monkeys, in primates, okay, in primates, apart from getting to lamina 5 and 6 and lamina 7, full of interneurons, they get directly to lamina 9 that is formed by motor neurons, alpha and gamma motor neurons, to extensors and flexors of appendicular muscles, and in particular, the muscles that are located, or the more distal portions of the limbs, okay? The fibers from the ventral corticospinal tract, they do mostly the same. They cross the midline and they end in lamina 5 and 6 and lamina 7, where there are interneurons. And some of them, they go straight to lamina 7 to activate motor neurons that reach muscles that are in the distal portions of the limbs. And this is for fine movements. Whereas the rubrospinal tract doesn't reach directly the motor neurons in lamina 9. Instead, they reach 5 and 7, 5, 6, and 7, where there are interneurons. So the information that is transmitted through the rubrospinal tract is not as direct as the corticospinal tract. It needs a lot of interneurons in order to reach the motor neurons. For the lateral tectotegmentospinal tract and the medial tectospinal tract, as you see, these axons end in lamina 6, 7, that are interneurons, and they don't go to lamina 9, because lamina 9 is formed by motor neurons to appendicular muscles. They end in lamina 8, that is formed by motor neurons to axial muscles. So these tracts control mostly not the limbs, but the axial muscles, okay? All right, we go back to histology, and you see this is the pyramidal decussation and the fibers that cross the midline and form the lateral corticospinal tract here, okay? So again, in this image, you see the decussation of the pyramids here. Here is the fibers that remain uncrossed, and here we have the fibers that are going to cross the midline. So you have the lateral corticospinal tract. And as you see, these fibers, here is the pyramidal decussation that cross the midline in order to form the lateral corticospinal tract. Here you have it in this image. You see we are at level of C2, as we see the gracial fascicle, the cunate fascicle that form the dorsal funiculus. And here you have the lateral corticospinal tract that is going to move now more laterally and becomes incorporated to the lateral funiculus. Again, you see these images. Right here, you have the dorsal horn, ventral horn. We are at the level of T3, T4, right? And what do we see here? This collection, these bunches of fibers that are more dense are the lateral corticospinal tract, and more ventrolaterally, we have the rubrospinal tract, okay? So, when we compare, again, the corticospinal tract with the rubrospinal tract, what do we see? We see that in primates, that the corticospinal tract is more important than the rubrospinal tract. Fibers reach directly lamina 9 of the ventral horn that is formed direct by motor neurons. There is a direct connection. Of course, it also ends in interneurons of lamina 5, 6, and 7 in order to reach lamina 9. Okay. However, in non-primates, this is when they have the corticospinal tract, this is the pathway they follow. They don't go as direct to lamina 9. In order to reach the lamina 9, they have to go through interneurons. Okay? So the animals that are non-primates, they do not have the fine movements that the primates have. For the rubrospinal tract, doesn't get direct to the lamina 9. It has to go through interneurons that are located in lamina 5, 6, and 7. Okay? So as we see in these papers, the rubrospinal output has been demonstrated to strengthen after unilateral corticospinal lesions, that is in humans. Because the rubrospinal tract is almost absent in humans. However, when the lesion affects the corticospinal tract, the rubrospinal tract takes its function. Again, we see that humans are believed to have more direct corticoneural synapses than any other species, consistent with observations that humans suffer more extensive loss of motility from lesions of the corticospinal tract than do other mammals. So in humans, the cortical projections are much more important in the movement than in our domestic animals. Again, we see about the corticospinal tract. Projection from the corticospinal tract not observed beyond the cervical level in goat, cow, deer, and sheep. And again, alternative descending spinal pathways such as the rubrospinal tract, reticulospinal tract, and vestibulospinal tracts are likely candidates in relation to body size of the reticulospinal tract in body, even and all goat ungulates. So, this corticospinal tract is not as well developed in our domestic animals. However, in dogs, it is this corticospinal tract, and in cats it's more developed than in dogs, because you just have to identify the abilities of in the movement of the limbs, of the hands and feet, right? To deduct the development of the corticospinal tract. So, what about the reticulospinal tracts, whether it's medullary or pontine? The medullary comes in the lateral funiculus, and the pontine goes in the ventral funiculus. And again, you see that how this tract ends in a lot of laminae, the dorsal horn, in the intermediate gray substance, and also in the ventral horn, ending in motor neurons, but most of them are gamma motor neurons, okay? And the medullary is descends bilaterally, and it is important in the inhibition of the flexor reflex interneurons in the dorsal horn. So it is important this tract to inhibit interneuron activity. So summarizing, we have the corticospinal tract that is controlling distal muscles of the limbs, the distal portions, okay? And this corticospinal tract is very important in the execution of voluntary movements. The rubrospinal tract is controlling proximal muscles of the limbs. Again, very important in voluntary movements. Remember that these tracts are not also excitatory, but are inhibitory over the interneurons. They have to stop the unwanted movements in order to have a correct movement. The tectospinal tract, the coordination here of the head, neck, and eyes, right? And the reticulospinal tract and vestibulospinal tracts here in the same color. They control axial muscles, the muscle tone, okay? They do not control the appendicular muscles, okay? Well, thank you very much.